All right, guys, we're going to talk a little bit today about the development of cell theory, how we discovered cells, and how that relates to modern biology. And so a lot of this it starts before this time. It starts around the 1500s with some guys that essentially put two lenses together and made the first microscopes. But where it really comes into play is a couple of guys back in the 1600s. One of the first of those is Robert Hooke. He's one of the original guys that helped found the Royal Society in London, where they really started to get into science and to discover things in the natural world around them. And in 1665, he publishes a book called Micrographia. And we see here a couple of pictures out of that book. And so a couple of really important things here. The first thing is he examines some cork, and he realizes that there are these little tiny sections that they call cells. And that's going to be obviously pretty important to us. The other really important thing here is that he does some amazing drawings. I mean, this is one of his drawings of a flea that he takes with his microscope. And you can see he's got it labeled in the different parts here. And he does a really amazing job. It's pretty cool because you can tell that he was obviously not just a scientist, but he had some pretty good artistic ability at the same time. The other guy that's pretty important to us is a guy named Anton van Leeuwenhoek. He's often referred to as the father of microbiology because he spends a lot of times, this was one of his first microscopes, and he spent a lot of time with microscopes, and essentially what makes him so famous is that he is looking at these things that look like basically little tiny animals. At one point they called them animalcules, that they were like what they pictured were little tiny animals. Now we know today that they're what we would call protozoa, so things like paramecia and stuff like that. So they're technically in a slightly different area than animals, but he did a ton of discovery of these types of things. It was really one of the forerunners of microbiology until we get to the next two guys, which would be Louis Pasteur, who you should know from pasteurization. He's really important in biology in a couple of ways, because not only do him and Robert Koch do an amazing job discovering things about bacteria. Koch is the guy that discovers anthrax. And he's a physician and does a lot of things to help to cure people of bacterial diseases. Pasteur, he's the guy that basically disproves the idea of spontaneous generation, the idea that life could come from non-life. Before his time, they thought that it was possible for life just to sort of spring up out of non-living things, and sort of the classic example of that was the idea that maggots come from dead meat. And so, as the meat was rotting, maggots would just appear. Now, we know today that that's not really the case, that flies have to go and lay eggs. That's where the maggots come from. But at the time, that was a pretty well-established, not well-established, but that was a pretty widespread theory at the time, that things just came from nothing. So all of these guys use some variety of microscope. And so we basically got three big varieties of microscopes here that are useful to us. Optical microscopes. Now, this is what you would probably use say in the classroom. Okay, the stuff that we're going to use most of the time are optical microscopes. And the way they work is basically you shine light through lenses and then you can view the objects. What is going to be cool about them, as opposed to some of the other stuff, is that in the case of optical microscopes, you can actually view things that are still alive, much like Leeuwenhoek did back in the day. The other two types, and since they're not really written here, let me write them out for you. The other two types of microscopes are electron microscopes. Okay, and there are two kinds. And the TEM here stands for tunneling electron microscope. And basically what it does is you fire a beam of electrons through a really thin slice of an object, and it goes through the other side, and then you're going to get an image. The other kind that's developed much later in the 20th century is called a scanning electron microscope. And what's really cool about the scanning electron microscope is it allows you to get three-dimensional images instead of sort of the flat images that you would get out of the tunneling or even out of the optical. And the scanning electron microscope is pretty cool in science terms, because this is where they're actually able to sort of pinpoint individual atoms for the first time. So that's pretty cool. Obviously, most of the time we're going to use optical, and the big advantage is here that optical, you can actually view living things. Tunneling and scanning electron microscopes, the things that you're viewing have to be dead, and they have to be basically set up and cut into these really thin slices in order to be able to use in those types of microscopes. So all of that work with microscopes leads us up to the idea of cell theory. We're going to be talking a lot about cells over the next couple of months. And so all of it basically boils down to, in some way, these 3 points of cell theory. And so the first one is that all living organisms are composed of one or more cells. Okay, that seems to make pretty good sense. The second one almost seems redundant. The second point of cell theory is that cells are the basic unit of structure and organization of all living things. But what this is saying is that, yes, we can say that all living organisms are composed of one or more cells. The second point here says that this is how organisms are actually organized, that they don't just have cells in them, that cells are what make them up, and cells are what supply all of the organization and structure of those different organisms. And so as we get into higher organisms, bacteria, one-celled organisms, archaea, and then as we get into the higher organisms, those cells get very specialized and get organized into systems and organs and stuff like that. But the cell is still the basic point. So like you have a liver, which is an organ and part of your digestive system, but the liver is made up of liver cells. Okay, and so at the smallest unit for living organisms, they're made of cells. The last one seems pretty wordy, but this goes back to Pasteur's point, is that cells arise only from pre-existing cells, okay, with cells passing copies of their genetic material on to the daughter cells. So that means life doesn't come from unlife in the biological sense, that there's no spontaneous generation, that there has to be a cell there in order to produce more cells. Now, we could get into all sorts of philosophical debates of like, okay, well, that's fine, well and good, so where do the first cells come from? Really not an issue that we can deal that much with in science, although there are scientists that are pretty interested in that and are trying to study what scientists would call the origins of life. But again, not really out of what we're going to deal with in this class. So for us, cell theory says that at the point that we're at, cells have to come from pre-existing cells. So what are the main types of cells? And we're going to dive into each of these in a lot more detail later on. But there are 2 main types of cells. You've got prokaryotic cells and eukaryotic cells. And the differences are that eukaryotic cells have fully defined organelles within them. Okay? Organelles are sort of like organs for the cell. Okay, so like we have organs in our body like our liver and our heart and our lungs, and they all perform different functions. And eukaryotic cells have all of these, you see all these little arrows here, pointing to all of these little different organelles that each have a very specific function within the cell. Prokaryotic cells are much simpler than that. Prokaryotic cells there's 2 main types of prokaryotic cells. So we got bacteria, okay, and archaea. Whoops, totally spelled that wrong. Okay, and bacteria and archaea, they don't have a nucleus in their cell, and they don't really have well-defined organelles. They've got some little different bits within them, but they're not really organelles. They're very, I don't want to say disorganized, but they're not organized in the strict organelle way of a eukaryotic cell. Now, these 2 different types of cells, it is proposed at one point that what happened is that, according to the endosymbiote theory, is that the way that eukaryotic cells came about, so like an animal cell like this right here, that the way those came about is that essentially at some point way in the distant past that a cell basically swallowed up another cell. And so then that prokaryotic cell that got swallowed up by another prokaryotic cell, it essentially became an organelle within that other cell. And so you can kind of see some of the reasons why that might be likely, because this right here is an example of mitochondria. And mitochondria are organelles within the cell that we'll talk about later. They're kind of similar in shape to prokaryotic cells, and not only that, but and because shape varies pretty widely, but what's really more interesting is that mitochondria actually have their own DNA that is separate from the nuclear DNA in a cell, and so that sort of lends credence to the idea that at one point these might have been different cells that were sort of swallowed up and enveloped and pulled in to form eukaryotic cells. The lady that came up with this idea, Lynn Margulis, super awesome scientist. I mean, she not only came up with this theory for biology and is sort of a big name in biology, but she's also a really big name in environmental science. She's the person that comes up that helps to go along and further the idea of the Gaia hypothesis as well.